This Week in Virology, the podcast about viruses, the kind that make you sick. From MicrobeTV, this is TWiV, This Week in Virology, episode 1297, recorded on February 13th, 2026. Yep, it's Friday. And I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Madison, New Jersey, Brianne Barker.
Hi. It's great to be here. Things are actually going pretty well on the Friday the 13th around here, so I guess I'll knock on some wood for that. And it's, above freezing again, which is novel recently. So it's 38 Fahrenheit or 3 Celsius, which is lovely compared to how it was, only a week ago.
Yeah, it's above freezing now during the day. It's really nice. It's 3C and sunny here in Chelsea. Also joining us from Western Massachusetts, Alan Dove.
Good to be here. And it is also tropical here in Western Massachusetts by comparison to the previous week. 36 Fahrenheit, 2C, just a couple of clouds. Actually, I can't even see them anymore. Mostly sunny. Lovely day.
Coincidental that it was Groundhog Day recently?
I don't know.
Probably is.
I feel like when we talk about the paper later, I can ask some questions about groundhogs. But
yes.
It's also, it's an association, right? It's not a clinic. It is not a double-blind, placebo-controlled observation. And from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Today, I am the Rich Condit equivalent in temperatures. That is, it's 40 degrees Fahrenheit here.
Wow.
And Celsius, that's 4.444444. So, and it's sunny, blue sky, really lovely.
Nice.
Wow. Is that weather moving our way or just going to
presumably? Yeah. Yeah. Usually you get my weather a little bit after I've had it.
I'm looking at the long range, through the next week, it's just 3 degrees, 6 degrees, 4 degrees, 5 degrees.
Oh.
So you said 40 degrees Fahrenheit, right?
Mm-hmm.
Yeah, that's
4.44 Celsius. Yeah.
Yeah, it's about the same.
Yeah.
6, 8 degrees, 5.
But you said, yeah.
Yeah, so it's still at night, it's freezing most of the time. But maybe spring is here soon. We'll see. If you enjoy these programs, someone wrote to me today and said, you have to stop discussing politics so that I can come back and listen again. And I felt like writing, how are you going to know if you stop listening? Dumbass. So stupid. Don't write to me and tell me not to talk about anything, okay? If you don't like it, you don't have to listen. We don't make anybody listen. What was I saying? If you enjoy these conversations, we'd love your support to make them.
And if you don't, buzz off.
Yeah, but if you don't, this is a program like other programs, right? Any kind of program, the people who make it decide. When they make a movie, they don't take a vote and decide what people want the plot to be, right? And if you don't like the movie, that's the end of it. Even with TV shows, they don't ask you. Podcasts probably don't ask you either. So it's okay if you don't like some of the programming. You can express but to threaten to leave is just, frankly, you're getting influenced by Orange Man. You're threatening things, okay? So stop it. If you would love to support us, go to I'm chastising you. Now
if you're not turned off yet.
Yeah, so it's okay. The thing I like about these podcasts is that they're our thing. We can do what we want. But if you enjoy them, and many of you do, I know that, go to microbe.tv/contribute, and you can help pay for them. They're free for everyone. You don't have to help pay. But if you, as someone has said years ago, if you enjoy things, you like to support them. The Rosenfeld Lab has positions. It has moved to Philadelphia from the FDA. Oh, my gosh, the FDA. What a mess. And they are looking for a postdoc and a tech to work on viral pathogenesis and immunobiology. They are interested in enteroviruses, cross-reactive antibody responses, the role in pathogenesis, trying to make animal models for these viruses, and also come up with new ways to deliver vaccines. if you're interested, you should email Amy. Her email is rosenfelda, it's one word, at mlhs.org. Now, let me just say another thing. Someone sent me an email about this job. I'm not the one with the job. Don't email me, okay? It's right here. In fact, the same person also emailed Amy at the FDA. I just said she's not at the FDA anymore. So this does not bode well for your employment, in my opinion.
Attention to detail is a key
Yes.
feature in laboratory work.
You got to follow instructions. But, it happens all the time. I say at the end of Daniel Griffin's thing, send your questions to Daniel at microbe.tv. Do you know how many people send them to Vincent at Microbe or TWiV at MicrobeTV? Or they say, I don't know where to send it to.
Yep.
The state of the world. Oh, my gosh. Anyway, why am I complaining so much? I don't know. It's been a tough week.
You're on a roll
today.
today.
May be the 13th. Today may be the 13th. The trains were all messed up this week. First of all, I get on the train at a little dinky suburban station with 2 tracks, right? One track goes east and one track goes west. And this week, everything has been on the western track. You know how people get messed up by like this morning I had to say to the lady, hey, come here. It's a very elderly lady. I know she can't run over the thing very quickly. I said, you better come here. The train is here. She said, but there's no sign. I said, well, it's on the app. And she said, oh, the app. And she started coming. Okay, so there was that. That's happening all
Also,
week.
just since you're speaking of trains, I looked it up. My train is electrified.
Okay, that's good. That's very good. And then the other thing that happened, Amtrak is always messing up. They had a wire issue, so they control the wires on the tracks. And they had a wire issue. They dropped us off in Newark and said, make your way into New York on your own because we're not going there. I find this offensive. You get people used to taking the train, right? Well, it turns out that there's another train there called Path.
Yeah.
And so I took that. But, they say, we will cross-honor your pass. There's no way. So I just walked through the gate like a criminal, okay? I did the same thing on the way home. And then the next day, it was still screwed up because of another issue. Some train, every day, either a train breaks down or they have overhead wire issues. You think the infrastructure is aging?
Yeah.
Anyway, that's my transportation rant. When I go to Europe, the trains run really well, right, Kathy?
Yes, they do.
Your experience?
Yeah.
What is it in Italy? The trains don't run on time. Wasn't that Mussolini's thing to get the trains to run on time?
The last European train I took was in Spain, and the train in Spain went right across the plain. It was lovely.
Oh, the train in Spain goes across the plain, yes. Okay, now it is just 11 minutes in, and we're going to do science. So don't give me hell that we talk about something else all the time, because that's 10 minutes out of 120 minutes. You do the math. It's not a lot. So, but first, some news, because there was big news this week.
There's science news, so this is also science.
News. It's all science, and it's discouraging. An article in SIDRAP, this is all over the place. FDA reverses course, refuses to review Moderna's application for a new mRNA flu vaccine. So back in August, when you do a clinical trial and you want to have a new product license, you don't just do it. It's not like watching a podcast and you have no feedback. You go to the FDA, you say, we want to do this. What do you think? And you go back and forth, right? Because this one, they spent $750 million on this clinical trial. That's what Daniel said anyway. I don't know if that's true.
40,000 people, so yeah.
Yeah,
40,000
that's probably
people.
about right.
So a flu mRNA vaccine, and they compared it to the current inactivated vaccine, the single dose, low dose, or whatever you call it, not the high dose, and it looked better.
A protocol that they had cleared with the FDA as discussed. Yeah.
Yeah. So then they go to, and when you submit your clinical trial, it's volumes of paper, like huge.
It's not even paper. They left off paper a number of years ago because it was just there were
too much.
Drug approvals would be convoys of semi-trucks going, from the headquarters of a drug company down the highway to DC and just like, movers with hand trucks. So yeah, it's handle electronically
Okay,
now.
well, that's good. Anyway,
Large amounts of data.
FDA said, and not FDA actually, Vinay Prasad on his own said, we're not going to consider it. And his committee at the FDA, which they supposed to do things by committee, they disagreed with that. They said we should look at it because they rarely not look at clinical trial results, and he decided one-handedly not to. And his excuse was they didn't compare it against the standard of care, which he claims is a high-dose vaccine. In my opinion, the standard of care is not the high dose; it's the regular dose vaccine. Right?
I smell a lawsuit.
You think so?
Oh God, if Moderna doesn't sue, I mean, I don't.
How do you sue the government, though?
Oh, same way you sue anybody else. Trump keeps losing lawsuits in court. This administration has been sued up and down, left and right.
Especially because in this article they talk about the previous conversations that Moderna and the FDA had about what would happen in the trial, and so if in a CIDRAP news article there is evidence that the FDA had indicated that this would be acceptable, I'm sure that there is much more concrete evidence in other places, and so it does seem like they would have.
And when we say conversations with the FDA, this is not a couple of people chatting over coffee at Starbucks. This is volumes of exchanges in writing. Yeah.
So Angie Rasmussen says the trial design they use is essentially the trial design that every single flu vaccine has used.
Yeah.
And the president said this does not make America healthy again.
Well, and that was a president of Moderna, right?
Yes, of Moderna.
Yeah.
Yes,
Moderna,
not the
yes.
yes.
Yeah, it's a good article. You can read it. But so I personally, as a scientist, would like to see this reviewed because I think we should modernize our flu vaccines.
Sure.
The technology is from the 30s. Come on. And if this thing did better than the existing vaccines, why not review it?
And even if it did the same, there are reasons why you might want to use a different manufacturing process rather than the ones that we currently use for some of our flu vaccines.
Yes, this in the case of a flu pandemic where we need fast manufacture and not egg-based allergy concerns and also drift concerns, this would be a big innovation, even if it's just equivalent.
Would they have the ability to try to get this licensed in other countries, or do they really? I mean, are they going forward with that? Do you think?
Well, yeah,
I hope so.
They have done that because, and they made a note that one of the countries is Germany, that they don't even use a high dose flu vaccine in Germany. So if they had tried it against high dose, you couldn't do that in Germany. So this is just crazy, and I think it's because RFK has this thing about mRNA vaccines. Remember, he canceled BARDA funding for mRNA, and I'm sure he said to Prasad, "Just can this. We don't want any mRNA vaccines." Just two people just having coffee and deciding, we're not going to do this.
Completely bypassing all the normal channels of government operation that are legally required. So not only do I expect Moderna to sue over this, I expect a whole lot of drug companies. The CIDRAP article mentions that this could throw a chill over the whole vaccine production industry. I would extend that further. I think this is chilling to the entire pharma industry, that you can spend $750 million on a clinical trial that you have discussed and that's been vetted by the FDA, and then one person there can arbitrarily and capriciously just throw it in the trash because they don't like the sound of it or something. That is just a massive risk for any drug approval.
Okay. That's a good point. Okay, then on the other side of the coin, we have I got an article in DW.com. I don't even know what that is. Anyway, Mehmet Oz's vaccine call is too little too late. So this week, Oz on CNN said, folks, you need to get measles vaccine. Right? It's the first time anyone in the administration has said that. And so this is being praised as something great. But all right, it's good that he said that. Maybe it'll make a difference. Maybe some people, I'm not sure how visible he is. maybe if he went on Oprah and said it, that would help, right? Because he used to do that all the time. But he said, take the vaccine, please. And this is a weird statement he made. He said, not all illnesses are equally dangerous, and not all people are equally susceptible to those illnesses. I didn't know that you could be susceptible to an illness, but measles is one disease where you should get your vaccine. Why would he say that? He's like, not all illnesses are equally dangerous. Why? Why not just say measles can be very bad, you should get your vaccine?
He's trying to hedge his political position so that he doesn't run crossways with the folks who are, some of whom we just talked about in this administration, who are staunchly anti-vax. He's trying to say, oh, well, but measles you should get.
I just wish that every time that the measles comes up and, it's in the popular media, that they'll talk about, well, and there's some danger of death and some danger of hospitalization. But I wish they would go further and talk about the fact that it puts all these kids at risk of amnesia to all the previous pathogens that they had immunity against. And that just doesn't come up.
Yeah. They could also always just make sure that they link the Roald Dahl piece where he talked about his daughter suffering from measles. It's a pretty dramatic piece that whenever I think about it, after reading it, I can never imagine not having a measles vaccine.
Yeah. So Oz is a doctor. He should take care of people. He shouldn't worry about getting fired, right?
I have conversations with Laura about this. She, my wife, for those who don't know, is a doctor. She actually did 3 years of surgery residency before switching to psychiatry. And she went to Columbia for medical school. And she sees Mehmet Oz as almost a tragic figure because as a heart surgeon, he was extraordinary. I mean, if you look at his surgical career, the guy could have just rested on those laurels and been regarded as one of the greats in the field. And then he fell in love with publicity and has gone off and done horrible damage to public health.
Yeah, it's seductive. That's the problem. Right. Well, I guess we're okay. We're glad at least that he said this. He's the only one that has the gumption to say it. But
and I really liked this article, which I looked up DW. It's Deutsche Welle or something like that. So something
Deutsche
German.
Yeah. Okay.
Deutsche Welle.
But I really like the graphics in this article. There's a nice map of measles cases in the U.S. and a nice graph of number of measles cases in the U.S. in different years that I definitely know that I will use again.
Oh, the map, when you hover over a country, it tells you where the country is.
Mm-hmm.
So there's one country that is in like Central Asia where it's eliminated. And I said, it's Turkmenistan.
Oh
my gosh. So, and look at all of Russia.
I wouldn't trade places with Turkmenistan for much else, but that's impressive.
No, I agree.
Yeah, the graphics are really good here.
Well, that's good. Belarus, Finland eliminated, Norway, even Iceland. But they don't have Greenland on the map. I'd like to know.
Well, it's part of Denmark.
Yeah.
So can we assume that? Denmark has it eliminated.
Yeah. So
does Portugal.
Greenland is
So,
Denmark.
I mean, I don't know why he didn't do it months ago when the thing was still pretty high, right? Why does he feel that now he has to do it? So I'm very suspicious. But as Alan said, he's just hedging something was bothering him that he should say something. Right?
Yeah.
Oh, here's the thing I wanted to point out. I read this somewhere else. He claimed that RFK said at the beginning of the measles outbreak, you should get your measles vaccine. What did he do? Whisper it in his arm while looking away from the mic? I mean, I never heard him say that.
Yeah, I don't remember him
So
saying
this
that.
is very suspicious.
I feel like
Dr.
if he
Oz.
said it with an awful lot of hedging.
Yeah.
His fingers were crossed behind his back.
It's in this article, they call him a former celebrity doctor. It's interesting.
Does that mean he's a former celebrity or a former doctor?
Yeah, I don't know. Maybe
I think he
both.
might, at this point, he might be more stung by the idea of former celebrity than former doctor.
He might, yeah. So he's no longer a working surgeon, right?
I doubt it. I can't imagine he would be.
How old is he?
Not young.
because you can do surgery. Oh, he's only 65.
Oh, okay.
So my dad was a thoracic surgeon. And by the time he was 60, he was just doing pacemakers, right? Because he didn't do the crack chest. Back then, they used to crack your chest open, right? But he stopped doing that. So I think at a certain point, there's a golden era, like maybe in your 50s when you're really good because you've done a lot, but then your motor skills decline or something. So he's 65. Yeah.
No, I think he's busy with his current job.
Okay. Let's do some papers. Got 2 papers for you today. And once again, I found a Journal of Virology article that I was interested in. And it is filamentous virions act as non-infectious interfering particles to modulate papillomavirus infection.
And the authors are, this is open access, by the way, Dominic van Bodegreven, Sridhapa Saha, Lilo Groen, Reinhard Kernbauer, Petra Dersch, and Mario Schellhaas. And affiliation list is on page 15, but at least they gave me a link. The team is from the University of Munster and coordinating University of Tübingen. It's an all-German group.
The first 2 authors are co-first authors.
Yes.
Kathy, you have a summary for us?
I do. So this is in our papillomavirus arc. We've predominantly been talking about those in the alpha virus or alpha papillomavirus genus. And there are 5 genera that infect humans. And today we're mostly going to be focusing on a virus that's in the beta genus. Anyway, papillomaviruses, as you remember, are small viruses, and they have a spherical capsid morphology and double-stranded DNA. So the alpha human papillomaviruses, or HPVs, as we tend to call them for short, include HPV 16 and HPV 18, and we'll hear more about HPV 16 in this paper as sort of a control. Those are 2 of the high-risk viruses that cause cancers of the cervix and elsewhere. They have a tropism primarily for mucosal tissue. And these are the HPV viruses that the vaccines are designed to protect against. And there are other alpha HPVs that are less oncogenic, and those are considered the low risk. But then this beta genus of HPVs is less well understood and less studied. They regularly infect the skin, but it is becoming clear that certain HPV types are now recognized to be in both mucosa and the skin. So 2 of the beta HPVs, 5 and 8, are potentially carcinogenic, and 5 is the one that we're going to hear mostly about in the paper today. And this potentially carcinogenic thing is that they can lead to a rare disease that can lead to squamous cell carcinomas of the skin that occurs in sun-exposed areas. Okay, and as you've heard a lot from Rich primarily, the life cycle of the papillomaviruses is complex. And as a result of that, that complicates growing the viruses in vitro. So in this paper, they use a technique that's called pseudoviruses. They produce these pseudoviruses as a surrogate system to study the initial steps in HPV infection. So these pseudoviruses have viral capsid proteins. They're virus-like particles in that sense, but the DNA that they have in them includes a reporter gene. So instead of asking for the virus to go through the replication cycle, etc., they're just asking for the virus to express this reporter gene, and then they can monitor the initial steps of the HPV infection. Okay, so as I said, the pseudoviruses are these VLPs, virus-like particles, and so the DNA they call pseudogenomes that have these other reporter gene in them as well. So now, stepping back to the big picture of some of the things they're going to address. For the alpha human papillomaviruses, there's a fair amount known about the entry. And there's a capsid protein called L1, and it binds to a cellular molecule called heparin sulfate proteoglycan, HSPG, and that will come up again during this paper. This binding to the HSPG also stabilizes a capsid conformation of the virions, and that's important for infection. And then a couple of steps ensue, and the virus is transferred to then what's they term an elusive secondary receptor complex. And by that, they mean they don't know what this secondary receptor complex is. So that's one of the puzzles in the field, and this paper is not going to solve that puzzle. But the thing that these authors are going to address is whether any of this holds true for the beta HPVs. And so evidence for some of these steps that are known for the alphas exists for the betas, but certain pieces of the data are partially inconsistent with that mechanism. And so there's not really good consensus, or wasn't, as to whether these HSPGs are used as the primary receptor for the beta HPVs. And so this paper is going to resolve some of that. They produced these pseudoviruses for this beta herpes virus HPV5. And they found that these pseudoviruses had low infectivity, especially compared to their control, the alpha virus HPV16. And they observed that their pseudovirus preparation had large numbers of filamentous instead of spherical particles. So this is interesting. And the two main findings of the paper then are that these filamentous particles acted like defective interfering particles. And so the more of them that were present, the less infective the pseudovirus preparation was. And so they were interfering with virus replication. And they were defective. They had this filamentous shape. And as we'll see, they're kind of a novel kind of defective interfering particle. This concept of DI, or defective interfering particles, has been known for decades. Previous inconsistencies about this HSPG debate are also resolved. And I'll cut to the chase and tell you that the data support that the HSPGs are required for the beta herpes viruses to bind to cells. And so we'll go now into more of how they do all these things.
Yeah, I've been looking at papillomavirus papers lately. I don't know why. There seem to be more of them, and it's really interesting. I wanted to learn more about it. So hence this.
They're spreading and popping up all over the place.
Oh, popping up all over the place. So we've talked mostly about the high-risk human papillomaviruses, right? There are 16 of them, and they're associated with a variety of anogenital and oropharyngeal malignancies. I didn't realize this number, 630,000 new annual cases of HPV-related cancers worldwide every year. That's a lot.
Yeah.
Major health burden. And so these are icosahedral particles. There's no envelope. I was making up we have an exam in my virology class on Monday. And one of the questions had there you go. There's an icosahedron.
There's the icosahedron for my virology class.
I got mine are made out of
next
magnets.
Monday. Yeah, this is also made out of magnets.
So I was saying...
I think the only icosahedron I have on hand is a D20 somewhere around here. But
I made the statement, what would happen if you wrapped this in a membrane? And so the TA said, it's too ambiguous. Why don't you say enclosed in a membrane? It's okay. That's fine. Wrapped. So these are icosahedral particles. No membrane. T equals 7. The triangulation number, 7 protein subunits per structural unit. And there's a major capsid protein, L1, which makes the capsid mainly. And in fact, the vaccine is just L1 produced in insect cells or yeast. And then there's an L2, there's a minor capsid protein. It's within the capsid, the inside of the capsid, apparently. And as Kathy said, they're grouped into five genera based on the L1 gene sequence. And the alphas like mucosal tissues. That's where the high-risk types are. And then we have the beta and the gammas that infect the skin. And one of them, as Kathy said, 5 and 8 are potentially carcinogenic. And so that's being looked at. And as Rich has said, this is very cool. They have a nice summary of the differentiation cycle. The viruses initially infect the mostly undifferentiated basal stem cells of squamous epithelia. and they have maintenance replication in those cells, very low copy numbers of the viral genome. And then as the cells differentiate into suprabasal spinous cells, that promotes replication of the viral DNA, and so they make a lot of DNA. And then as the cells terminally differentiate into keratinocytes at the top, the genome replication slows, and you have more production of the L1 and L2 proteins, which now allows you to make virus particles. So you have enough DNA by the time you get up there, and then you release the particles. But all this makes it hard to grow them in cells because you have this differentiation-dependent replication, right? So that's why they use pseudoviruses as sort of a surrogate for infection. Virus-like particles that have a reporter gene inside of them. Do these pseudoviruses have any of the viral genome or just reporter? Do you remember? I don't remember.
I didn't look at that detail.
Yeah, but they do have a reporter. But the fact that they're pseudoviruses means that they can't carry out the whole reproductive cycle, right? Okay, so Kathy told you about entry and what's been done about that. It's actually quite interesting. For alpha HPVs, the L1 protein of the capsid binds to heparan sulfate proteoglycan, specifically the glycosaminoglycan portion, and this interaction stabilizes a particular conformation of the capsid that's needed for infection, and then this activation of the capsid by interaction allows cleavage of L1 by a protease, a secreted cellular protease called kallikrein, which I remember from my graduate school days. Everybody was talking about kallikrein. And it reminds me of the things that blue whales eat. What are they? Krill.
Krill. That's
Do you know that blue whales eat a ton of krill a day?
Yeah.
Tons of
Makes
krill.
sense.
Where do they find it all? Oh my gosh. Anyway, that
in their
cleavage
mouths.
by krill allows externalization of the L2 end terminus from the lumen, right? It goes from the inside to the outside. And then that is cleaved by furins, which is also secreted, which decreases the affinity for the heparan sulfate proteoglycans, and then transfers the virus to an elusive, as Kathy said, an elusive secondary receptor, which one day when they find it, we will do it here on TWiV, because that could be.
It will not elude us.
And there are receptors for that, but we don't need to talk about them here. So then the cells take up the virus by a novel endocytic mechanism, which I've never heard about, called wash-mediated endocytosis. It's all going through the wash, I guess.
Yeah, I also noticed that and had never heard of it and made the decision to not learn more about it right now.
But what it does, it brings the virus to the Golgi, okay? And then from there, the DNA gets into the nucleus. So it says that it gets in the nucleus after the envelope breaks down during mitosis. I didn't know that either.
Yeah, at that point, I had already spent enough of my morning watching videos of monkeys eating squirrels. So.
Okay, so also, as Kathy had said, there's some inconsistencies about entry and whether heparan sulfate proteoglycans are a receptor for the beta HPVs like 5.
Right, so the alpha HPVs, what Vincent just gave is the description for all this information we've got about the alpha HPVs, which we have, other than the elusive second receptor, we have a pretty good outline of.
Now, because the alphas bind HSPGs, their binding can be inhibited by soluble heparin, right, which will bind the virus and block its attachment. But the betas are not efficiently blocked by this, and this is going to be solved in this paper. Nobody really understood why, and they assumed it meant that it's binding a different receptor.
Well, but there's evidence in both directions, which is the inconsistency they talk about. You can see that HPV5 binds heparan sulfate, but also you can't stop it. It's not effectively competed, and so there's this back and forth of does it, doesn't it?
Right. All right. So they're fooling around with pseudoviruses. And the HPVs, when they purify them on velocity gradients in a centrifuge. I have a picture of the tube here. The HPV-16s, you see one, what we call in the field one band, which is certainly jargon, but what else are we going to call it, right?
It's a stripe across the tube. It's a band.
Stripe across the tube, what we call a band. And if I just said to you, it's a band, you wouldn't know what I meant, right? Because a band to people is something else. It's usually
with
something around.
sousaphones and drums.
Well, no,
Sousaphone.
it could be interpreted. You have banded, snakes or whatever. There's
Or squirrels.
There are
We have
bands
banded
or squirrels,
squirrels.
or a raccoon has a banded tail.
Mhm.
But anyway, in this, the HPV-5, perhaps they saw two bands. So the upper one has the infectious particles in it. And they're wondering what the other one.
I love this. I love this experiment because it's a case where they're actually telling you how they ended up doing this work.
Yeah.
I mean, somebody was doing a prep on virus and they looked at the tube and they said,
Well,
"That's
why do we have
weird.
this? Yeah.
What is this?" And then they found that one of the bands was infectious virus. So what's the other one?
It's an interesting point that I can, if a graduate student is doing this, the PI, depending on the PI's metal, they could say, just do your experiment and ignore it. Don't pay any attention to
Right.
it.
It's
Or,
not some contaminant. It's some crud in the tube.
don't
Just
worry
get
about
done.
it. Don't
Yeah.
worry about it. Just go and get the answer that we want. And then others will say,
Hmm.
wonder what it is. And you have to do that. You have to be curious. That's how you discover things.
And it's the interesting thing about it is if you purify alpha pseudoviruses, you don't have this second band. So it's something distinct in the beta pseudoviruses.
Yeah, and they show pictures of this really nicely in
Yes.
the paper for people who are curious.
So they noticed that the HPV-5s infect HeLa cells less efficiently than HPV-16. So they said maybe it's this lower band. Let's see what's going on there. And so they looked at the number of genome equivalents in a preparation, right? The number of pseudogenome containing particles, because these are pseudoviruses, by PCR. And so relative to L1, the capsid protein, which they used to standardize, the HPV-5 contained about 4.5-fold fewer genomes than HPV-16. But this only accounts for a small part of the reduced infectivity that I just told you about. And so they end up looking at this lower band and they find it's not infectious and it's got filament. And they say, this is very funny. We haven't told you it's filamentous yet, but then they call it the filamentous enriched HPV-5 for reasons that'll be apparent in the next paragraph.
Yes.
Because they couldn't figure out how to give it the name without, giving you
The spoiler. Yes.
The spoiler. Thank you. It's very funny. So this, these filamentous enriched HPV-5, they have no pseudogenome incorporation and reduced amounts of L2 protein and histones. So the L1s have histones on them. All right. So then they wanted to look at these by electron microscopy, right? Because they've learned a few things about them, but now they want to see what they look like. And both HPV-5 and 16 have spherical particles, as you expect, 55 to 60 nanometers in diameter. But both populations of 5, both bands have filamentous particles that are not present in the 16 preparations.
Brianne, for those who are listening
there's a
and not watching, she's holding up a filamentous and
There's
an
a filament.
icosahedral.
Yes,
And when we,
I have
Yeah.
built both with magnets for my classes, and they were sitting on
Yeah, I
my
got 'em.
desk.
They're on the
So-
shelf behind me, yeah.
and when we refer to spherical particles, that's just the description of the electron micrograph. Those are actually the icosahedrons. They look like spheres in the micrograph.
They actually are spherical, but they're built with icosahedral.
They're built with
symmetry.
icosahedral, yeah.
Yeah.
Symmetry. Yeah.
So these the spheres are the filaments are 30 nanometers in diameter, so about half the diameter of the particle, and 200 nanometers up to several microns, so they can be pretty long. And there are pictures of them here. So basically, HPV thinks it's a filovirus now. So they think these filamentous particles are non-infectious, although they don't have bona fide wild type viruses, as you'll see later. Okay, so what's going on here? They did first of all, they have to separate the two better than they did, because in the previous centrifugation, both bands had filamentous particles. So they come up with a centrifugation method using potassium bromide cushions. Oh boy, that's a jargon if I ever heard of one. What is that? Something you put on your couch?
Well, no. Once again, I think the jargon in this case is actually kind of sensible, because it is a cushion.
Yeah, once you
at
know
the bottom
what
of the tube,
it means,
and it's functioning as that.
Sure,
And-
but at first glance, right?
Yes, right.
And then the way I think of it is, I've never heard of those kind of cushions, but I know cesium chloride cushions.
Yes.
And
Yes.
so the cesium
Or
and the
sucrose cushions,
potassium
yeah.
and the bromide and the chloride, it
Yep.
all makes sense. Okay, got it. It's-
All right. So then, when they separate, now they have spherically enriched HPV-5, because it's not zero, and it's much more infectious when you take away these filamentous particles. So they say that infectivity increase correlates with the removal of the filamentous particles. So, in other words, the filamentous particles are impairing the infectivity of the spherical particles. And so they say, well, these sound like defective interfering particles. As Kathy said, those have been known for decades with many viruses. DI particles, as they're known, they typically have a deletion in the genome, which maintains the reading frame, and they interfere with the replication of the non-defective virus in mixed infections. So the DI on their own can't make new particles typically, but they depend on the wild type virus, and they also interfere with the wild type virus. Okay, so then they start looking at binding, so to see how they're being defective particles. So they first do an experiment to show that these sulfated glycosaminoglycans are important for binding. So they can remove these cell surface heparin sulfate or chondroitin sulfates by treating cells with either heparinase or chondroitinase. And then you try and infect the cells. And in fact, if you remove chondroitin sulfate, you impair HPV-5 infectivity a little bit, but a lot. Removal of heparin sulfate strongly reduces HPV-5 infection. So heparin sulfate is important. The inhibitory competition for binding with heparin, which is a soluble heparin sulfate analog, requires much higher concentrations for HPV-5, and it's inefficient at even high concentrations. But at low concentrations, you get more efficient infections compared to HPV-16. So that's very weird. And so, and then if you spherically enrich HPV-5 with reduced amounts of filamentous particles, they have a dose-dependent decrease in infection upon heparin challenge, right? So if you've reduced the amount of filamentous particles by this purification method, now you have heparin sulfate sensitivity, whereas if you don't take away the filamentous particles, it's not so sensitive.
Right. So you take away the filamentous particles, and now HPV-5 behaves like HPV-16.
16. And so they say, well, maybe the non-infectious filamentous particles somehow interfere with heparin's impairment of the infection by the spherical particles, right? That's the idea.
Which sort of makes sense if, they're basically soaking up all the heparin.
That's right. They're soaking up the heparin, and we're going to find out an explanation for that in a moment. I mean, they're long, right? A lot of binding sites, right? That's avidity. Keep that in mind. All right. So they say maybe these filamentous particles engage the glycosaminoglycans more efficiently, and therefore heparin can't compete with the binding of the spherical particles. So you could do that by either having higher affinity or avidity of the filamentous towards heparin sulfate and heparin.
So you've alluded to the difference in terms between affinity and avidity, but I bet to many people those words mean almost the same thing.
Right.
So how would you explain that?
So the affinity is a measure of the binding strength of, let's say, heparin, one molecule of heparin to a virus particle. The avidity is the sum of all the bindings of many molecules of heparin bound to the particle. Better put with an antibody, right? A single antibody bound to an antigen has an affinity, but if you have 5 antibodies bound, then it has a higher avidity.
Yeah, and so the affinity is like a single binding site, and avidity is kind of the
Multiple,
combined multiple binding sites, and it's usually not just the sum of the affinity. There are sort of additional factors that make avidity usually be a bit higher than the sum of the affinities.
Anyway, they do binding assays, and they find that the affinity is the same for spherical of heparin sulfate is the same for spherical and filamentous particles, but the avidity is different. So that's why, and there are lots more binding sites on the filamentous particles.
Yes.
So they're a high avidity decoy.
Yes, Brienne's showing the long
Yes.
filamentous
The long
particle.
filamentous,
More sites.
All right. So let's see. Next, this is very interesting how they word this. So now they have a model, right? And they say to challenge this model in which the particles act as a high avidity decoy, the filamentous particle, we did this experiment, which I thought was very interesting
Yeah.
because it frames it for you.
Right.
Instead of saying we then tested whether,
Right.
and you're not quite sure why, right?
And I found that the to be a little bit ambiguous, so I crossed it out and wrote our to challenge our emerging
That's better.
model.
Yes.
That's better. Yes. Now, I'm not sure. Certain people don't write that way though, but I like that better than the yeah. All right, so they test whether titrating in filamentous HPV-5 VLPs would confer higher competitive effects than spherical, and so they add an increasing amount of either the spherically enriched or 16, and they test infectivity with or without the heparin challenge, and they find addition of the filamentous much more prominently reduced infection of the spherical than addition of the same molarity of just spherically enriched HPV-5, so better binding to cells via HSPGs. And then if they put heparin in, only the filamentous enriched led to increased infectivity in the presence of heparin. So preferred binding of filamentous particles to heparin. They're better binders, as we've seen, than the spherical particles. They did this with HPV-16 also, and they find the filamentous particles of 5 inhibit HPV-16 infectivity because they're binding to the same receptor as binding to heparin, right? And there's a control, interesting control they looked at, a debris fraction, which has pieces, right?
Yes.
Subviral structures and pentamers, and this doesn't reduce infection. So you need to have the capsid. You need to have the filament, in fact.
They even have the picture of the debris. I love
Yes.
that.
Yeah. They also did an experiment where they they labeled the particles, the filamentous particle with it's spherical, and the filamentous particles with 2 fluorophores, right? Two molecules that are fluoresce different colors under UV light, and they can actually look at binding directly to the cells under a microscope, and they find the same effect. In the presence of heparin, the filamentous particle give a stronger decrease in binding, so they engage heparin more efficiently than the spherical particles. Now, this is in a lab, right, with HPV-5. What about in the real world? So they make this note that cottontail rabbit papillomaviruses have been observed to form filamentous particles before. And so they want to know how general this is. So they get some warts from an immunocompromised patient, and they find lots of spherical particles, but no filamentous HPV-5 particles in them. So these are 5. These are HPV-5 positive. And so they say, maybe this is a lab thing. but rabbits are not a lab thing, right? So maybe somewhere in the world there are filamentous particles in the wild.
Yeah, I mean, maybe if when you actually get to a point of having warts, you for whatever reason have a predominance of the spherical particles, but maybe in someone who has who's not, who has an inapparent infection, perhaps
that could
their infection is predominantly filamentous, but how would you ever
Yeah,
find
good luck
that?
finding that if they're inapparent.
Yeah, right. So then they look at some other samples. They looked at a set of alpha and beta human papillomavirus preparations, and they find filamentous particles in alpha HPV31, beta HPV5, which we've already done, and HPV23. That's another beta, whereas alpha 6, 16, and 18 were devoid of filamentous particles. So it seems to be specific to whatever the number is, right? It's not alpha or beta specific.
Right.
And maybe you would be able to, if they looked some more or elsewhere, maybe you'd find them. So now this is cool because you have identified the receptor for HPV5 by discovering these filamentous particles, right? So it has implications for entry. It's not just a weird lab artifact.
Right.
But it is, in a way, just an artifact. I mean, and they pursued it, and then it gave him this interesting result.
I mean, it's something readily observable in the lab, but
right?
Whether or not it's in a host, who knows?
Right.
Because the one person is not enough to really say.
But the fact that they pursued this artifact in the lab got them to a point where they could figure out what the contradiction was in the earlier results, and now we know something about the virus in the wild as well. It binds heparin sulfate truly as its initial receptor.
You know what I think happened? They did their gradient, they saw this lower band, and they say, what's that? And they put it right under the EM. And they saw that it was filamentous, and that's why they did everything.
If they had easy access to EM, then that's certainly
I think
what I would do.
That's what I think, because if you saw these filamentous particles, you would go, oh, wow, what are these? And then you initiate.
But
that's my guess.
I bet that they took the two bands and they put them through the infectivity assay.
Yeah,
right.
I think that would be before the EM.
They probably do that first, yeah. But before all the other competitions and everything, I bet they looked at it.
Well, you could also imagine that they could think about how well they wanted to purify their sample. And did they care about being precise about separating those two bands, or did they care about lumping them together? And if they tried to do the individual bands versus sort of lumped together, they pretty quickly would figure out the difference in infectivity.
Yeah.
So they make the point in the discussion. They say, we think this is aberrant particle formation, but cottontail rabbit in vivo has these filamentous, so we can't rule it out.
It's not necessarily just a pseudovirus.
Yeah. Mm-hmm. So they point out that this is a novel defective interferon particle because most of the other ones interfere with the replication of the virus, and this one is interfering with attachment. They say a feature that, to our knowledge, is unique among viruses. And what's interesting, though, is what makes it filamentous, right? Because normally the proteins self-assemble into a nice spherical capsid. So what drives it? And they think that maybe their in vitro system for making these pseudogenomes, they're overproducing the capsid protein, and maybe that is influencing the fidelity of the an overabundance of capsimeres maybe are leading to this.
So they describe it as they're overexpressing these the L1 protein and having so many L1 proteins around. Maybe it's forcing it to go into some other conformation instead of making the nice icosahedral shape. It forces them to make these linear filaments.
It would be,
the vaccines, the HPV vaccines, are made by producing L1 in various culture systems, and I'm sure people have looked at them under the EM. There's no filamentous.
They have
particles.
Because I actually used that paper in class, so I know the paper and I know the EMs in that paper.
But it would be interesting to see if you could alter the conditions of, say, HPV-16 pseudovirus production in such a way that you could force it to do this, to see if it's alpha or beta, if it's beta specific.
Yeah, So it must be, the expression must be different in the vaccine production and this to
Right.
lead to this. But it would be. I mean, I'm sure they're immunogenic. Also, I'm not sure they would be harmful. I mean, if you saw these, because the Hep B vaccine makes these elongated particles as well, and they're okay. Nobody worries about that. You get the spherical ones and you get the elongated ones. So
You've got
not
the
a bad
antigen,
thing.
and it's
Yeah, it's enough. Okay. So that's filamentous HPV. All right, now we have a Nature article, which is entitled Transmission of MPXV from Fire-Footed Rope Squirrels to Sooty Mangabeys.
How could you not click on a title like that?
I mean, I saw it and I didn't even read it. I said, "We're gonna
Fire-footed
do this
rope
paper."
squirrels, you got me. I'm in.
Well, I
Yes.
looked at the map and I said, "Yeah, we got to do this
Yeah,
paper."
they
Because we have an MPX arc, not as
yes.
not
So
recently. but-
MPX, the virus formerly known as monkeypox, and this group, whole bunch of people here. So the co-first authors, this is also open access. Co-first authors, Carm Riutford Fay, Jasmine Schlotterbeck, and then co-senior authors Sebastian Calvignac Spencer, Ariane Dux, Olivia Patrono, and Fabian Lindertz. And the group is from Helmholtz Center for Infection Research in Greifswald, Germany, Centre Suisse de Recherche Scientifique en Côte d'Ivoire in Abidjan, Côte d'Ivoire, Université Pelaforo Gon Coulibaly Korhogo in Korhogo, Côte d'Ivoire. I apologize for butchering pronunciations here. Seckenberg Museum for Natural History in Gorlitz, Germany. Friedrich Loeffler Institute in Reims, Germany, Technische Universität Dresden in Zittau, the Queen Mary University of London, CNRS University of Lyon, German Primate Center in Göttingen, Université Félix Houphouët-Boigny d'Abidjan Cocody in Abidjan, and University Medicine Greifswald in Germany. And the Côte d'Ivoire connection is because that's where this paper takes place.
I have
And it's
summary,
just a
Kathy.
cool paper, too.
It is so cool.
Yeah. So Vincent also gave us a News and Views article, and I think they're both paywalled, the News and Views and the primary article.
Primary paper is not paywalled.
okay. Okay, good. The News and Views, I think, I had to do an extra step. So,
Yeah,
but
I did
anyway,
too. Yeah.
and the title of it is the shortest of all summaries. Squirrels could be a reservoir for Mpox.
Yes.
Okay, so as Alan said, Mpox is now the disease, and monkeypox virus is the virus, and the monkey part of it is really a misnomer, as we're going to find out or reconfirm with this paper. And so the authors report on an outbreak of this virus, MPXV, in a group of wild sooty mangabeys in the Thai National Forest. And they have access to this vast database of samples, longitudinal samples, and from various places in the Thai National Forest, spanning many years. It includes specimens from rodents and primates. It includes behavioral data, which is amazing. And the authors perform sequence analyses and other studies, and they conclude that there's likely transmission of the virus from this fire-footed rope squirrel. And so the News and Views gives a really nice summary and points out the caveats. And in my reading of the News and Views, it's almost too harsh of pointing out of the caveats. The authors also allude to most of those caveats, which we'll discuss. And the authors even say that there should be more research and continued surveillance because the involvement of several host species seems likely. So they're not trying to say that this fire-footed rope squirrel is the only reservoir.
It may be fire-footed, but this is not the smoking squirrel.
Yeah, well, they have a sentence in the paper that I thought was just really sort of key. They say, and so they basically say that some of their data hints at an exceptional case of real-time detection of a cross-species transmission event. Yeah. And that to me is the key thing. They they don't exactly have, every detail of that cross-species transmission event, but they come pretty close. And just in terms of, probabilities, the rareness of being able to see events like what they're able to show here is absolutely phenomenal.
Yeah,
And
we don't have anything close for
No.
SARS-CoV-2
No.
or SARS-1. No.
Right.
And well, we'll get to this too, but in the supplementary data, I mean, when we talk about seeing it happen, you see it happen.
Yeah.
It's
Yep.
like, oh my gosh,
very
it's
good.
there.
So-
and I
so-
just wanted to point out for listeners, the Thai National Forest is TAI. It's not in Thailand. It's in Cote d'Ivoire.
And it's the same one that is sometimes referenced in the naming of Ebola viruses.
Yes.
Thai forest Ebola virus.
And just the reason why this work is important is that with habitat changes, there's a change kind of in the overall structure of the mammals that are present, tending towards smaller mammals due to disruption of habitat and so forth, fewer primates potentially. But there's still consumption of them as bushmeat and as subsistence. And so that increases the possibility of further transmission. And so being aware of these squirrels and other rodents as potential reservoir hosts is really important.
Yeah, they sort of talk about that in a few different places in the paper. And I know that there was a talk at a conference a couple of years ago that sort of hinted at some of these things as well, in terms of exactly which animals should we be paying close attention to, in terms of conservation, but also in terms of, bushmeat and in terms of consumption. And I spent some time down a rabbit hole of some of their citations. And there's some really interesting stuff where they talk about that in some of the papers that they reference.
Yeah.
So, monkeypox infection is a zoonotic infection, right? It comes from some reservoir, which we're going to learn about here. But so there are multiple spillovers from whatever that reservoir is into humans. And more recently, we've seen a lot of them. And then there's also sustained human-to-human transmission. We've talked about that in one of the papers not too long ago. And so it's a public health emergency of international concern, according to the WHO. And clearly, we need to identify the reservoir. We know bats are reservoirs for SARS-CoV-1 and 2, but we don't know exactly what the reservoir is for monkeypox virus. And you need to know to manage the spillover risk, right?
And they have another thing another sentence that I particularly liked in the sort of earlier part of this paper was they give a really specific definition of what they mean as reservoir.
Yes.
And I absolutely am planning to, take this sentence from this paper where they say, we define a reservoir as a natural host in which the virus can circulate permanently and from which transmission to another host is possible and documented. And so the thing to remember about a reservoir is that it's not just some animal that can host the virus. It is one where you have sort of long-term circulation. Sometimes that is thought to mean perhaps lack of disease or longer-term persistence, and that's something that they sort of hint at in a couple places here in terms of whether or not what they're looking at is a reservoir. But the sort of idea of that permanent circulation, as opposed to what they'll talk about in, say, the sooty mangabeys, where it looks like basically these organisms, the mangabeys, got infected and sort of had a disease outbreak. It wasn't like a permanent circulation in their population.
And also these reservoirs can transmit to humans, in which case it's a zoonosis, but they can also transmit to other animals.
Yep.
Right? And we don't learn about that as much because, for obvious reasons, right? When humans get infected, we know it. And so they say, according to this definition, no monkeypox virus reservoir has yet been identified. Okay, so monkeypox virus was first isolated from wildlife in 1985 from a Thomas's rope squirrel captured in DRC, tested positive. So supporting the idea that this one and other African rodents are natural hosts. And other studies have supported this analysis. For example, PCR of museum specimens have found monkeypox virus DNA in 5 rope squirrel species. And more recently, screening small mammals in DRC reported near complete genomes of the virus in a Thomas's rope squirrel, another squirrel, and a soft-furred mouse. Nice names they give. Almost as good as trees. And also overlap analyses of the niches of 99 different African mammals and human mpox cases similarly point to squirrels as a most likely source of human infection, okay? But we don't have any direct evidence of transmission to humans or any other hosts. Now, an interesting event in the U.S., there was a monkeypox outbreak in the USA in 2003, which was all the human cases were linked to prairie dogs, pet prairie dogs sold by one distributor who housed them with several African rodent species imported from Ghana in a single shipment. So I feel like Sherlock Holmes here. This evidence is getting overwhelming. It's just all adding up.
The 2 suspects crossed in this room. Yes.
And among these rodents, 3 of them tested positive for monkeypox virus. So we have giant pouched rats, rope squirrels. Again, rope squirrels show up, and dormice tested positive. Okay.
Feed your head.
Then they went back to Ghana and looked, and they found members of the same species, either seropositive or PCR positive, okay, for orthopox virus. And so, so the prairie dog is probably an incidental host, between the rodents and humans.
They just popped
but
up.
It doesn't tell us much about zoonotic transmission.
Right. You sort of imagine there was a shipment of all of these things together, and only one of them had to be positive and spread to the others. So we have sort of three suspects, but that doesn't tell you which of the 3 suspects it might be.
Right.
So the reason it's called monkeypox virus is because captive non-human primates were associated with its discovery, and then so that it was a misnomer because non-human primates don't appear to be the reservoir, and they have also done long-term monitoring at the Thai Chimpanzee Project, which is the site of this study, the TCP, Thai Chimpanzee Project, in Thai National Park in Côte d'Ivoire. Monkeypox virus also affects non-human primates. So now we have more insight in the ecology of this. They found monkeypox virus in a dead sooty mangabey, and later found 3 independent monkeypox virus outbreaks in 2017 and 2018 that were in distinct groups of Western chimps living in the same forest. And the source of these we don't know at the time, but in January of 2023, they started beginning to observe clinical signs compatible with monkeypox virus infection in some infants from a habituated mangabey group, which consisted of 80 individuals in this same location.
And
so
I want to call out the wildlife observation, the extraordinary amount of wildlife observation that went into this discovery.
Yeah.
These are not captive animals. They're in a park. The habituated mangabeys, I gather, are associated with humans, but that's like, the raccoons at the local park. They're still wild animals, and they have had I gather, decades-long, consistently funded project of wildlife biologists watching and studying these animals, the chimps, the sooty mangabeys, and they were able to document this timeline that is, as you'll see, absolutely critical to what they're about to discover.
Collecting samples.
Yes, and collecting
urine,
samples. Right.
Etc.
And
this similar work done a long time ago in the chimpanzees in Cameroon identified the origin of HIV, right?
Yep.
Yeah.
getting in there and collecting thousands of
It's
chimp
not all
samples.
about the sexy molecular biology in the lab. You got to get people out there with binoculars.
Yeah, well, and I was really, I was impressed by a lot of things in this, but there is a figure that actually shows baby mangabeys and adult mangabeys with pox lesions. And so to be able to, and they, to be able to actually make those kind of observations and make observations about what's going on with the clinical observations of these mangabeys is pretty impressive. I don't think I've ever seen a mangabey with disease that was obvious of any kind.
Yeah, so this group of mangabeys, they point out they're stable. They form a stable, cohesive matrilocal social group. So they're interactive. They develop 30% develop skin lesions over the next 12 weeks following late January 2023. And so they did an outbreak investigation to find where these sooty mangabeys got this infection. And they have pictures of these mangabeys with, rashes all over the body or on the head. This tiny baby here, he's got rashes on his face. Poor guy. Anyway, at the end of January, one infant mangabey got lesions on the forehead, the back of the head, the chest, and the legs, got lethargic, didn't want to eat anymore, and eventually died 48 hours later. And by early March, 5 other infants had developed similar lesions, and 3 of them died. They also saw a milder form of the disease with a rash, fewer lesions, and this affected 20 other mangabeys of all age groups. And in all of these, the lesions evolved to crusts and then eventually scabs. And overall, so the disease went through the group until the end of April of 23. They had 26 out of 80, or 32% of the mangabeys developed at least one visible skin lesion and 4 deaths. So they sent veterinarians in, which they note were wearing personal protective equipment and following strict biosafety protocols to do necropsies on 3 of the 4 infants. The 4th, they said, was never found. I guess something ate it. So first they tested the necropsy samples by PCR, and they identified viral DNA in all the main organs. And then they looked at the whole group, and they analyzed 170 fecal samples collected during the outbreak. That's the period during which they had clinical signs. And they found monkeypox virus DNA in 36 fecal samples from 19 individuals. 7 of them were symptomatic and 12 were asymptomatic. Of the 19, 14 were mothers of symptomatic infants, and only 6 of them developed lesions. They didn't detect any virus in 89 fecal samples that were collected after clinical signs were gone. So they say this shows that monkeypox virus caused this outbreak and probably infected more people than we know, more mangabeys than we know. I'm so human-centric.
Yes.
Because we see subclinical infections here. Okay, so next they're going to do some genome sequencing and see how this fits in with other monkeypox virus isolates. So they get 2 near-complete genomes from a skin sample and a fecal sample, and they are identical across 92% of the genome. They also got a partial genome from a necropsy sample of a different individual, and they were identical as well. They put the virus as a clade 2A virus, and the sequencing tells us that the outbreak was a result of transmission involving the same local reservoir. Okay, so then what do you do next? They look at rodents and shrews, 694 of them, found dead inside and around this Thai National Primate Center between 2019 and 2024. So this is stuff that's ongoing.
So they're anytime they find a body, I guess they take samples and freeze them, and they've got this archive. Again, amazing, consistent wildlife observation.
So they found one monkeypox virus positive fire-footed rope squirrel found dead in November of 2022, 12 weeks before the outbreak in the mangabeys and 3 kilometers south of their territory. And so all organs of the squirrel and oral and nasal swabs had high viral loads, and they could isolate infectious virus from skin, lung, spleen, and liver of the animal. Must not have been dead for very long, I guess. It's amazing.
Yeah.
Right. And they point out they don't know what killed it, right? It could have been the virus or it could have been something else. They just don't know.
Yeah, and that's where they're sort of making that point about reservoir and not.
Yes.
Yeah.
I mean, this rope squirrel could have gotten it from some other thing, which
Yeah.
is the reservoir.
Yeah.
We just don't know. So they say that, the growing body of historical, spatial, and ecological evidence, including what we have here, supports the idea that rope squirrels are a natural host of monkeypox virus.
Now, this
So they-
particular rope squirrel, 3 kilometers south of these mangabeys, is not thought to be the one that gave it
No,
to these
it's
mangabeys because that is beyond the ranges of both of these animals.
Right. But we will see how rope squirrels
Yes.
factor in. So they sequenced the genome of this rope squirrel, infected rope squirrel, and 181,000 bases, nearly identical to the genomes from the mangabeys. Okay, so that's pretty
The
striking.
viral genomes, yes.
The viral genome. Now, there's only 81%, and in the commentary they say, "Oh, that's not enough to prove anything." It looks damn good to me, 80.
Mhm.
But yeah, there could be very substantial differences elsewhere that would say it's not from that squirrel. So
I thought
let's
it
keep
was
that in mind.
92%, but anyway, it was 92%.
That's
that you mentioned
92%.
earlier. Yeah.
Sorry, 80. Yeah, the lung sequence was covered only 81, so it's 92%.
Yeah.
Okay,
They were.
so they do some molecular clock analysis, and they say it would take 13 or 20 months before 2 epidemiologically linked sequences diverged at a single genomic site. And so they figure the time to the most recent common ancestor of the mangabey and squirrel viruses that they have here is 2021. Okay. But they say if the events that led to the infection and the outbreak in mangabeys were epidemiologically related, one would expect the 2 viral genomes to be identical.
But
It's
you've
so
got
close.
it right. So what they've proven is that it's in these fire-footed squirrels, these rope
Yes.
squirrels, very close, not close enough for that particular sample to have been the source. But in that same time frame, the mangabeys have this outbreak a little while later, and the timing on your molecular clock is within the window. So all the Venn diagram is all lining up, and
Right.
there are these rope squirrels all around that area.
So then they say, we're going to strengthen this hypothesis. And so they test 114 fecal samples from the mangabey group in the 16 weeks before the outbreak. They were collected before the outbreak. And they're looking for MPX DNA. They find 10 MPX positive fecal samples. And they were in 7 mangabeys that were asymptomatic at the time of detection, including the mother of the first infant to show clinical signs. The 3 earliest positive samples were consecutively obtained from the same individual on 3 days in December. So in other words, the virus entered the group. Maybe this is the index
We
case,
may have found monkey 0, yes.
and then circulated in the group for nearly 2 months. Okay. Now, they say mangabeys are known to hunt small mammals, including in this TNP. So they looked in an archive and they found a video from 2025 showing a mangabey catching a squirrel, and an older video from 2014 showing a mangabey eating a fire-footed rope squirrel.
And by all means, go to the supplementary data and click on those links.
Yeah,
Yeah.
the video, the photos.
They are eye-opening.
I mean, it's amazing that they. that they have that.
The one catching the squirrel goes up to a tree and kind of looks at it, reaches into a burrow and yanks out a squirrel and then starts eating it. And the one eating the squirrel is holding an already dead squirrel. And the way they eat it, I mean, it's like they're just grabbing a hot dog. They just start chewing on the squirrel's head until the head is empty, and then they start
Nature is
chewing on the
tough.
rest of the squirrel.
Nature is tough.
Yeah.
All right, so now that they know that the mangabeys eat squirrels, they say, let's look for squirrel DNA in the fecal samples that we have. And so they identified DNA perfectly matching the mitochondrial genome of the fire-footed rope squirrel in 2 fecal samples. So squirrels are not only eaten, but the group fed on the species on 2 times in the weeks before the outbreak.
This is the smoking dung.
And then it gets even more smoking.
Even more striking, they found one of the 2 samples containing squirrel DNA was also the first monkeypox virus positive fecal sample of the suspected index case. So there you go. It's like SARS-CoV-2 in the market, right? Commingling with DNA from susceptible animals. Here you have in the mangabey that got sick first, you have virus and you have
this is even better because it's one monkey poop that gives you this whole story.
Yeah.
There's the squirrel, there's the virus.
And this monkey ate a squirrel, and so that can't be the same squirrel as the other squirrel you looked at because that one had not been eaten. So that's another way that you know that other squirrel was not the
this is where the sentence that was cited earlier, this hints at an exceptional case of real-time detection of a cross-species transmission event which led to the group-wide monkeypox outbreak. But then they say because the squirrel detected in this fecal sample cannot be the same as the one found dead more than a month earlier, right, outside the home range, this co-detection constitutes further independent proof of monkeypox virus circulation in the rope squirrels in the area during this period. So the one they found dead 3 kilometers south, it can't be the same one that this monkey ate. Right?
The monkeys do not have refrigerators. Yeah.
So they say this. We think that the hypothesis that the squirrels are a reservoir is plausible. It seems plausible. I think that's pretty safe, right?
Yeah.
Then they go on to talk about the fact that bushmeat is an important source of protein here. Large-bodied mammals are declining in these regions because of habitat destruction and hunting, and people are starting to eat smaller mammals, including rodents, and they sell squirrels at urban outdoor markets. And squirrels live close to villages. They can live in fragmented habitats.
In fact, they cite, so they've got a database of where is it? They highlighted this somewhere.
Carrion flies.
Yeah. So on reanalyzing a mammal generic metabarcoding data set derived from carrion flies collected along a gradient from pristine forest to the surrounding villages at the same site. And they found the fire-footed rope squirrels were in secondary forests and plantations. So they're using the carrion flies as a bioassay to determine what lives and dies in the different areas. Somebody did this sampling of the carrion flies and did, barcoding of all the mammals the carrion flies were feeding on. That's awesome. Yeah. What a resource.
It's great.
Yes.
It's a great resource.
So from that, they know that these fire-footed rope squirrels are preferentially in the disturbed area, the secondary habitat, the close to the areas closer to the humans.
That sort of reminds me of the white-footed mice and possum story with Lyme disease and ticks.
Yes.
They say that squirrels are commonly trapped by people, including children, and so there's an opportunity for zoonotic transmission. Yeah. So they're trying to build a case that not only could the squirrel be a reservoir, but there's also opportunities for zoonotic transmission.
Yeah. And I just, whenever we're talking about bushmeat, I always want to point out, this is what we call game when white people are eating it. And descended from somebody from a family that consumed a lot of squirrel, and having tasted it myself, I'm not passing judgment on people for eating perfectly good squirrel meat, but this is something that you might want to, have some kind of an education campaign. Be careful when you're cleaning squirrel because they carry viruses, maybe.
Yeah, one of the references, and I don't know if I can find it now, but one of the references actually talks about surveying people and to find out what which types of bushmeat they're eating and why and what reasons they have. And I've actually seen some talks on that at bat conferences as well. And so I think that those types of things are also important about finding out what people are eating and maybe convincing them maybe they should try something else, or actually having some of these conversations.
And maybe figuring out what in the food system, has people eating squirrel. If it's something that is socially prestigious to do, which I kind of doubt, then you may have a hard time combating that. If it's something that's out of need, then maybe you could address that problem in the food system and get people to stop eating the squirrels.
So I'm wondering, because they do kind of make a distinction, or they list both in a sentence, subsistence hunting and bushmeat hunting, and what their definition of the difference would be, or...
My understanding of that, as I've seen the terms used elsewhere, is when people refer to bushmeat, they're often referring to commercially sold meat in the markets, whereas subsistence is, your family has a farm and you also have the kids trap squirrels so you can supplement your meal.
Okay.
So
my
obviously
assumption is that that's what they meant.
Okay.
Obviously this work is important to develop a program to mitigate, spillover risk, but they end up with an interesting sentence. They write, the example of TNP, the Thai Forest Primate Center, simultaneously shows the direct link between a rodent host and a spillover host, and that both may be sources of human infection, suggesting that a focus on squirrel consumption alone would be misguided.
Yes.
And I've just realized it is reference number 10, if someone is really interested in this, that I was reading that talked about these issues.
Yes.
Saving rodents, losing primates.
Yes.
Why we need tailored bushmeat management strategies. Okay.
It's never just one thing. It's never simple.
That's right. Okay, we just have 2 emails today, so let's read them. Alan, can you take that first one?
Sure. John writes, Doctors TWiV, looking forward to hearing some details in upcoming epitopes on Prasad's rejection. This is referring to the mRNA flu vaccine, I believe. Reading between the lines of what little detail I've seen, my guess is that since, of course, you can't create your own control vaccine, Moderna picked as a control an existing vaccine that had the best efficacy toward the flu strain closest to the one they were targeting with theirs. That would seem legit. Not legit would be taking the vaccine that has had the best efficacy ever to any flu strain, yet not targeted against the strain expected to be circulating, and that Moderna designed theirs against. In fewer words, I expect that Prasad doesn't appreciate the moving target that flu is and how difficult this makes things. But as things stand right now, I see at least one good outcome already. From their own court, CDC has tacitly acknowledged that the proper control or placebo is not saline.
That's right.
Good point.
Other good news, February is now 40% over. A few days ago, there was a one-day-only special in Greater Braddock. The high was 57 Fahrenheit, 14 C. That ended a record-tying 17 days in a row with temps below freezing. Today went back to below 0 C all day long. Thank you, John. I actually, my understanding from the SIDRAP article is that the rationalization that Prasad gave was that Moderna should have used high-dose flu vaccine as their control instead of regular dose. But in fact, the FDA had already agreed that regular dose was perfectly appropriate control.
But certainly not saline as their control.
No, certainly not saline. So very good point.
This is the problem when you make conclusions that are just wrong.
Yeah.
Right? That saline has to be the control. And so Prasad is not even thinking about that.
Nope.
Not that this is based on any rational thinking, but when you're there's some saying here, when you're messing with lies, you're going to get more lies out of
Yes.
it, right? Brianne, can you take the other one?
Sure. Paul writes, Dear TWiV team, this is a story of defending patients' rights and the choice to receive immunization. I am the attending physician at a nursing home where 4 residents had a court-appointed guardian who declined consent for any vaccination due to a rigid anti-vaccination position. These individuals had no relative or close friend able to serve as their guardian, so a public guardian had been appointed, someone who makes a profession of guardianship. There was state vaccine registry evidence that these individuals had previously received numerous immunizations, indicating a previous wish to be vaccinated, and 3 of them expressed a current preference to receive immunization, even though cognitively impaired. A lawyer and I petitioned the probate court requesting that a new guardian be named for these individuals because the current guardian was not performing the duty of consenting to what the individual would likely want, not what the guardian would want. After a 2-hour hearing in which the public guardian in question, without legal representation, did not make any argument that her personal anti-vaccination position should prevail over the evidence that these individuals wanted to be vaccinated, the judge appointed new guardians for each person. The related topic of childhood vaccines being consented to or declined by parents raises the similar ethical issue of parents' freedom to make decisions which place the vulnerable child at risk. I recall the case of Ethan Lindenberger, who when he turned 18 defied his anti-vax mother to get immunizations, and gives a link. Thanks for all your work. I'm a MicrobeTV financial supporter. Paul.
Wow. It's a really
Good
scary
work, Paul.
Good job, Paul.
Essential work.
Yes.
I mean, can you imagine a guardian who's anti-vaxxer?
Yeah.
This is so scary.
Yeah, guardianship can be problematic, especially when you have a government appointed, as he puts it, people who are professional guardians.
Professional guardians who are anti-vax. I mean, they should screen you to be not. I mean, that's not.
Well, I mean, anti-vax is only one of the possibilities. There are other things that can happen in that situation that are
that's the
not
thing
good
on
for
our
the
minds
patient.
now, right?
Yeah.
So, yeah, good on you, Paul, for doing that, getting that fixed, and also for supporting MicrobeTV.
Yes.
Yes,
Thank
thank
you
you.
so much. Thanks for the story too. It's great.
Mm-hmm.
Let's do some picks. Brianne, what do you have this week?
I'm kind of sticking with the book/TV arc. I'd heard about this book series for a long time, but had never gotten to it on my list of books to read. I'd also heard that there was a TV series, and recently just decided to check it out. And so this is a TV series called Silo. The book series is called Silo. The first book is called Wool. And I inhaled the Silo TV series very, very, very quickly and became sort of a bit of an evangelist for it almost. So there are 2 seasons that are out right now, and those 2 seasons represent the first book in this series, Wool. There are 2 more books in the series, and there are 2 more seasons of the show coming up. But right now, there are the 2 seasons of the show that are relevant to the first book. And so I'm now reading the first book, Wool, which corresponds to the shows that I've watched. I'm really excited when I finish reading Wool to get on to the next book because it gets to parts of the story I haven't seen yet, although they do change quite a few details between the books and the show. I do like the show quite a bit, actually. So the basic idea here is that there are 10,000 people who live in an underground bunker. They have no idea why they live in an underground bunker or what happened to result in their living there. They have a society there. The worst offense you can make is to say you want to go outside. And outside, they understand to be highly toxic and not a good place to be. And so this seems to be some kind of, far future dystopian Earth kind of setting. And so here we are, seeing the society inside the silo and learning about what's going on in the silo and what might be keeping them there and just sort of what the setup is like. And there are it's enough like other dystopian things that I've read that it's sort of or other seen that it kind of seemed like a story I knew, and yet there were many places where I was surprised by sort of things taking a turn that are sort of not canonical and that I hadn't necessarily expected. So I have just enjoyed this TV series, but then this book is sort of my main thing I've been reading right now, and I cannot wait to get to the next book and learn more about why they ended up in the silo.
Kind of sounds a little Matrixy.
It sounds a little Matrixy, although
People living underground, right? The last of the humans.
Yeah,
And the earth above is a wreck, right?
I don't want to do too much spoiler in terms of what I have already learned about both the world outside and what's going on with the people underground. But I could see that from that description.
I mean, it's got to be a really big silo for 10,000 people, right?
Yes.
It's not just like
it
a
has
corn silo.
No, it has 150 floors. With people
Okay.
living on different floors, and there's sort of a class system based on which floor you live in, and different floors. Some floors are devoted to farms, and some are apartments, and some are offices, and some are mechanical, and some are chemical labs.
Do people get vaccinated in this society?
They don't talk about it. So that's a good question, but I would certainly hope so.
It's an interesting idea because now I presume there's no animals there, right?
There are some rabbits, and
Yeah,
I
but
think there
there's-
might be a few other animals.
Okay, so there are no red
Yeah.
fire squirrels, so
Right.
zoonotic risk is lower than above.
Yes.
Right.
Interesting. Okay, that's cool. I might get to that one too. You have a good track record so far in what I've read.
Well, it's good to hear. I'm glad you're enjoying them.
Kathy, what do you have for us?
Well, I didn't pick a book. I picked a story that I saw back in November, and when I went to the links, they were now broken. So I had to go back to the primary articles by way of PubMed, and that helped me find the second paper. But the main one is entitled Ancient RNA Expression Profiles from the Extinct Woolly Mammoth. Okay, this is in Cell, so it's open access. And the dogma is that, shortly after an organism dies, DNA is degraded, and the RNA is degraded, and so forget trying to get RNA. But this challenges that because they have isolated RNA, and they started with permafrost samples of 10 woolly mammoths, 10 to 50,000 years old, and they did muscle and skin sampling. And they did this RNA profiling, which basically allowed them to work out some RNA pathways. In one mammoth nicknamed Yucca, the RNA was complete enough to tell that it was involved in building and maintaining muscle cells. And then the senior author says, well, that was kind of a bit boring, but it points out the way for future studies to find RNA from extinct animals' hair follicles. And so he said, if so, we may finally learn which genes made woolly mammoths woolly. And then the earlier paper, it was 2 months before that, and it's actually DNA-based, entitled Ancient Host-Associated Microbes Obtained from Mammoth Remains. And so from that, they identify, Pasteurella, Streptococcus, Erysipelathrix, and Actinobacillus. So various sequences from bacteria that could be pathogenic. And they have something from the oldest mammoth sample representing the oldest authenticated host-associated microbial DNA to date. So I'm not sure if your TWiV folks know that yet, Vincent, but might be of interest. So
It's cool.
These are really cool.
yeah.
Yes.
This is awesome.
Could be a twin paper, yeah.
Mm-hmm.
Yes.
This week in
Definitely.
mammoths.
Yeah. Alan, what do you have?
Okay, so I was torn between two different books by this author and ended up picking this one. This is Ubik by Philip K. Dick. If you don't know who Philip K. Dick is, you have certainly encountered his stories before. You can look him up. And a little while ago, I thought, of all the sci-fi I've read, I have only read like a couple of short stories of Philip K. Dick. I really ought to read at least one or two of his novels. So I initially picked up The Three Stigmata of Palmer Eldritch, which is awesome. And I was thinking, I should pick that for TWiV. But this book, Ubik, takes all of Philip K. Dick's big ideas and loads them onto an express train and sends it careening off the rails. It is an absolute screaming acid trip of a book. And if you read sci-fi, you got to read this because it's also been hugely influential.
Yeah, it's a classic.
It is a classic for a reason. What he does with it is just astonishing. It's very difficult to summarize. The main character is a guy who runs a company they have anti-psychics who protect corporate interests from having their secrets stolen by psychics. So it's got a little bit of an Inception aspect to it, but they go to what they think is going to be a lucrative job, but it's a setup, and they get blown up and injured, and then things start getting really, really weird, and their spaceship ages backwards and eventually becomes a Model T, and they're trying to track down the way to get out of this, and there's something called Ubik, which what is God, and is he available in a spray can? It's just it's an experience.
His books usually are.
Yeah,
he's great. Do androids dream of electric sheep?
Yes, became Blade Runner.
Yep.
Yeah. I read a lot of his short stories, and they are amazing.
Yeah.
Oh my gosh.
Yeah.
This one I haven't read, so I just bought it.
So
I'm gonna read it.
he also has some. I can't remember the name of the book, but it became Minority Report.
Yes.
And the first book of his that I ever read was The Man in the High Castle.
Ah, yes.
Which is another really famous
Another good one.
That also became
Cool.
a show afterwards.
I have a book also by Neil deGrasse Tyson. It's called Starry Messenger, Cosmic Perspectives on Civilization. And so the title Starry Messenger comes from Galileo's book Sidereus Nuncius, which he published in 1610, right? He said, Earth is around the sun, it's not the center of the universe, and people didn't like him for that. He went through a lot of trouble because of that. But this one is like a modern day answer. Let me read from his website. He's writing about Galileo. These first ever cosmic perspectives were ego checks on our self-importance, messages from the stars forcing people to rethink our relationships to one another, to Earth, and to the cosmos. We otherwise risk believing the world revolves around us and our opinions. As an antidote, Starry Messenger offers ways to allocate our emotional and intellectual energies that reconcile with the biology, chemistry, and physics of the known universe. So he writes about war, politics, religion, truth, beauty, gender, race, And I got this idea from him that, or I think he said it, what was it? Having to do with gender. Well, maybe I actually coined it myself. The Republicans want medical freedom, right? Yet they will not allow people to be the gender they want to be.
Right.
What the hell?
It's hypocrisy. Yeah.
Anyway, because he was talking about that in the book in some other vein, and I just thought, anyway, so he's got a very interesting approach to life in the world. He's got opinions, and he said he went to Las Vegas once because he likes to go and gamble, and he was in the airport, and he said they had no science books. So he said, apparently they don't want you to read why you're a fool to go gamble. Because he said if you knew some statistics and probability, you would never
wouldn't
gamble.
be there. Yeah.
Yeah. Anyway, I enjoyed it a lot. It's also a very quick read. I've been reading a couple of his other books as well. So I find that nice. It's good.
This looks good.
It's easy. He was faculty at Princeton for a while. I didn't know that. He got his PhD. I forgot where, but he did something maybe postgraduate at Columbia.
Was PhD at Cornell or was undergrad at Cornell? He's got some affiliation with Cornell. And then he ran the Hayden Planetarium at the Natural History Museum. Still does.
Yeah.
Okay, right.
So he was being selected for a jury, and the judge was talking to him, and the judge said to him, "Do you have any questions?" And he said, "Yeah, I don't know how you can convict someone on the observations of one person. You have to have more than one witness. It doesn't work otherwise." And so the judge,
"You're excused,"
said to the jury, Yes, eventually you got. Yeah, I said I was home before the end of the day. But the judge goes to the jury. Do you agree with what he just said? And he paraphrased it, and he got it wrong. He didn't actually say what. And so one of the jurors said, "That's not what he said." He said, "At least someone was listening."
Mhm.
It's really good stuff like that. I really like. Okay, we have a listener pick from Beth. After listening to your recent picks of Swedish crime fiction, I had to add my recommendations for another country's crime fiction. Iceland has some terrific crime writers. The most well-known are Yrsa Sigurðardóttir, Sigurðardóttir, and Ragnar Jónasson, but there are many others. I particularly like Yrsa. She has a series of excellent crime thrillers featuring a lawyer named Thora Gudmundsdóttir. She has written good horror fiction as well. I'm not much of a horror fan, but I really liked I Remember You, a horror story, and The Undesired. More psychological tension than blood and gore. Highly recommended. Beth. Okay, that's cool. I didn't know Iceland had good, I guess every country has good crime fiction, right? Except Greenland.
Well, they're not a country. They
I mean,
probably have,
Denmark does, so I'm sure
yeah,
Greenland can take some
Sure.
ownership.
What is Greenland called?
Greenland. It's part of
Part
Denmark.
of Denmark?
Is it a what Puerto Rico is called to the U.S.? What is it a
Are they a territory or are they just a province of? Or are they like Curacao?
let's see.
Now,
what
of course,
it
you
is.
get gazillion.
It's an
news
unincorporated
stories.
U.S. territory.
Yeah, Puerto Rico is a special situation.
It
Should
is an
be a state.
autonomous territory within the Kingdom of Denmark.
There you go.
I think Puerto Rico should be a state, frankly.
Yes, I agree.
And so should many other things. That
But
we
it's not
have,
up to
it'll-
me.
No, because it's going to be they'll all be democratic,
Well,
and so nobody.
And it's actually now a self-governing nation as of 2009 under the Self-Government Act.
Ah,
Sorry,
You know,
Kathy?
well, maybe if Puerto Rico became a state, it would be the 51st state, and
Yes.
therefore Canada couldn't be
Couldn't be.
the 51st state. Just thinking.
Right. It's right. And so we play with politics here on TWiV. So don't get bent out of shape, folks. Relax. All right. That's it for TWiV 1297. Show notes, microbe.tv/TWiV. You can send us questions, comments, pics. TWiV@microbe.tv. But don't tell us not to talk about politics. We hardly do. 2 hours tonight. Very little politics. And if you'd like to help support these shows, you can contribute at microbe.tv/contribute. Kathy Spindler is Professor Emerita at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Brianne Barker is at Drew University, BioProf Barker on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot.
Alan Dove is at alandove.com and turbidplaque.com. Thank you, Alan.
thank you. It's always a pleasure.
So you still do both of those websites? Is that right?
Yes, I still. So
You write on both.
alandove.com is now my more serious website because I found people were going to that by default.
Yeah.
And so that's like kind of my professional face. Turbidplaque.com is all my goofy side projects. So if you
Okay.
want to read about weird, nerdy side projects, go to Turbid Plaque. If you want to, just get some sci-fi book reviews and some thoughts about recent stories I've done, then go to Alan Dove.
Okay. I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.